Can somebody confirm you can hear me?
I can hear you.
Thank you. Can you see me?
No.
All right. My camera doesn't seem to be working, and I'm not sure what to do about it, but it does work if I share my screen. So for the moment, we're just going to go with it. Why did that break the page? I don't remember that being the case. Let's start schedule. Let's leave it there. All right. Before we begin, are there any questions? Should be seeing my screen now. Don't think we finished chapter 14. Um, Let me state that there's a few things you need to finish because not everyone has done that. I think we're further than that.
You did 13 instead last week.
Oh, 13. That's right. Stay up there. I don't think we finished chapter 13. Yeah. All right. Let me show the syllabus here. So today is November 25th, and we are supposed to be, oops, wrong month, sorry. I don't remember this breaking before, but today is November 25th, and we should be starting chapter 13 today. We're not quite there yet. Let me remind you in the lab that the unknown project is due this Saturday, November 29th, and the unknown notes pages will close on Friday, November 28th. If you ask for a test on Friday, make sure it is a 1 day test because otherwise you will not get the results in time. Oh, and on your unknown notes page, the unknown notes pages will be graded and it'll count as a regular lab grade, meaning for 5 points. So make sure you have your last results up in your unknown notes page, and you should also state what your unknown bacteria is. You do not need to give a table stating how you figured it out, but you should state what your unknown bacteria is. Not everyone has done that, and maybe they didn't realize that the unknown notes page is graded, but it is. You should also have on the unknown notes pages everything I have stated in the unknown notes page instructions. Any question about any of that? Let me remind you, there's no class on Thanksgiving. This is Thursday, so happy Thanksgiving. Any questions on anything? All right, let's go back to chapter 13. I had that we were talking about retroviruses, and we discussed how they're a positive single strand RNA genome, and they're unusual in that they have reverse transcriptase in them, which takes the single strand RNA in 2 steps, makes double strand DNA. First, it makes single strand DNA, and then the reverse transcriptase makes a double strand DNA. which the double-strand DNA can then be transcribed. By the way, it integrates into the host cell's DNA, and it can make uh, messenger RNA, which is positive strand, single strand RNA, which can serve as messenger RNA or as genomic RNA. And you can translate the messenger RNA into the capsid proteins, which get together with the genomic RNA to make the new progeny virions, which will bud out of the cell, usually out of the cell membrane for retroviruses. Any questions about retrovirus and how it multiplies? Retroviruses, when they um, combine with the cell membrane, they have an envelope, and normally that envelope fuses with the cell membrane, allowing the retrovirus to be brought into the cell. The reverse transcriptase is inside the virus, one copy of the protein, and it'll take the viral genomic RNA to make double-strand DNA and two steps. And then that viral DNA will move into the nucleus and integrate into the host cell's DNA. And this integration is fairly random, meaning in different cells, it can happen in different locations in the DNA, different chromosomes. We call that a provirus, as we spoke before. And that provirus can be expressed to make messenger RNA, which is also genomic RNA, and will move into the cytoplasm to make the viral DNA, which will then, some of it will go on the cell membrane, and some of it will make the capsids of the capsid code of the virus, which will get together with the viral genome to make the new virions, which will then bud out of the cell, usually out of the cell membrane for retroviruses. I'm not sure of all species, but at least the ones that I know of bud out of the cell membrane. Any questions about retroviruses? Remember, retroviruses are a RNA virus that actually replicates in the nucleus. It is the one exception of viruses that replicates in the nucleus, even though it's an RNA virus. Let's briefly talk about coronaviruses. Coronaviruses are a single positive strand RNA virus. It is an envelope virus, and there you're actually seeing it here. This is my, what do you call it, login page for the microbiology class. I had this slide up there many, many years before COVID-19 was ever in existence. It is a coronavirus, probably one that causes the human cold, and I just kept it after COVID-19 came out because it's obviously important. COVID-19 actually looks like this because it is a coronavirus. And as I stated, coronaviruses were well known to humans long before COVID-19 came out because they are the second most common cause of the human cold. Coronaviruses, besides causing human cold and COVID-19, also cause SARS and MERS. SARS stands for Severe Acute Respiratory Syndrome, MERS, Middle East Respiratory Syndrome. Both of these diseases are similar to COVID-19, meaning Severe Acute Respiratory Syndrome. MERS, this is the highest mortality rate, where about 36% of humans who come down with MERS die. But fortunately, MERS does not spread patient to patient because the virus doesn't spread people to people. It tends to spread from camels to people. And I assume it also spreads from camel to camel. COVID-19 when it first came out, had a mortality rate between 0.2% to 3%. But after, gosh, what is it, Omicron came about, the mortality rate has halved. So the mortality rate now of COVID-19 is about 0.1%, 0.1% to 1.5%. And COVID-19, as well as SARS, spread much more easily than the flu, with one person likely to infect 1 to 2.5 other people. And that is why COVID-19, we've never been able to get rid of it in the human population. SARS has actually hit the human population twice, and the first time was in 2003, and the other time was a few years later. I don't remember when that was. Fortunately, SARS, the medical profession was able to stop SARS from spreading. And the reason for that is that SARS, almost all cases of people who get it, they have symptoms, signs and symptoms. Whereas COVID-19, about 50% of young people and children, meaning young adults and children, will be asymptomatic. And that's why COVID-19 actually came to the United States. If you remember, Trump put a moratorium on anyone coming from China. I don't know if he did it to anywhere else in the world, but uh, and then they were checking people coming into the airport. But the trouble with COVID-19 was about 50% of young people were asymptomatic. So it did come into the United States. And then we had it spreading all over the world. COVID-19 does have another name. COVID-19 actually is co for, let's see, covariant. So coronavirus, that's what the COV stands for. The ID means it was identified in 2019, where the 19 comes from. So that's what COVID-19 stands for. It does have a scientific name. Its scientific name is a severe acute respiratory syndrome, coronavirus 2, or SARS coronavirus, coronavirus 2, which is so similar to SARS that not even scientific personnel tend to call it SARS covariant 2 or severe acute respiratory syndrome coronavirus 2. COVID-19 is closely related to bat coronavirus and likely originated from a recombination of two bat coronavirus and a pangolin coronavirus. How we know this was we sequenced the original COVID-19 variant and discovered that it was very similar to two bat coronavirus and then the pangolin coronavirus. The pangolin coronavirus only makes up about 10% of COVID-19, but it's an important part. The pangolin encodes the spikes on the coronavirus. Let me end that and recorrect that. Oops, that's not right. Oh well, let me go ahead and start it again. The bat coronaviruses code for the spikes, which are the reason why COVID-19 is so easily spread among humans. That the spikes are what causes the variant to easily spread and infect human cells. There likely was an intermediate that introduced COVID-19 to humans. It may have been a pangolin, although there's something else like a jaw dog or something like that which they think may have been the intermediate. We don't really know which animal was the intermediate. It may have been only one animal carrying COVID-19, and then a human picked up COVID-19, and then the variant started spreading among the humans. Any question about any of that? They think COVID-19 came in a Chinese animal market, which the Chinese used to have a lot of markets with a bunch of exotic animals in them. And having these animals in close proximity to large numbers of people who came into the animal market made it much more likely for COVID-19 to jump from an animal to a human and then start spreading among humans. And this has actually happened more than just with COVID-19. That is actually how SARS twice entered the human population. The Chinese have now realized that having these animal markets with all these exotic animals in close proximity to large numbers of people is a health concern. And so they've gotten rid of at least all the exotic animals like pangolins. If you don't know what a pangolin is, it's a spiny anteater. And so when you go to the Chinese markets now, you'll find more regular animals like chickens and pigs. I don't know what else, rabbits maybe. Um, you won't find pangolins and whatever else animals like lizards and I don't know, other anteaters and maybe monkeys. You won't find all those exotic animals. And the reason is the Chinese realize that it's just too risky. They've had three very serious diseases, SARS twice, which I didn't mention it, but that disease killed about 10% of humans when they got infected. And it was a good thing that the medical profession was able to stop it from spreading, because otherwise it would have spread around the whole world like COVID-19, and there'd be many more deaths than COVID-19. Any questions about any of that? All right, COVID-19 is primarily spread from droplets from coughs or sneezes or from singing within a range of about 6 feet, but it may spread by indirect contact via a contaminated surface, but it does tend to spread primarily person to person through droplets. Usually breathed in. It's rarely picked up by hands and then carried to the mucous membranes in the face, but it can survive for 3 days on plastic or metal surfaces or for a day on cardboard or paper. After about a day on cardboard or paper, the virus, the outer membrane tends to dry out, and then the virus is non-infective. COVID-19 symptoms are flu-like. The statement here that the shortness of breath and deaths are more common than the flu. The runny nose and the sneezing are less common. That's not true so much anymore. Ever since Omicron came out, COVID-19 has been much more like a flu-like disease. Omicron was a covariant that was actually COVID-19 merged with a coronavirus that causes the human cold. And that made Omicron spread a little more quickly, but it also reduced the mortality rate of COVID-19 by 50%. It's still lethal, but the mortality rate has dropped about 50%. And all other variants of COVID-19 have all been descendants of Omicron. The mortality rate of COVID-19 increases with age, with the ill health of the patient. The mortality rate is more common among men and more common among Blacks and Hispanics than whites and Asians. This has been seen now, at least in 3 countries, I'm not sure if anyone's looked further, but it's been seen in Mexico, in the United States, where it's first seen, and in Canada. That blacks and Hispanics, for whatever reason, it's a more severe disease. The incubation period of COVID 19, you get signs or symptoms between 2 to 14 days after first exposure, but realize some patients are asymptomatic, with about 50% of young patients being asymptomatic. The actual death rate in the United States before Omicron came out was about 1.8% of people getting COVID 19 died. That rate has now dropped to about half that. Prevention, use correct hand washing, keep your distance from people, especially when they're coughing, and don't touch one's face. If you're sick, you should wear a mask so you don't spread COVID 19 to someone else. But it's best to stay home and cough into your elbow or tissue. Any questions about COVID 19? All right, let's talk about viruses and cancer. Some human cancers, about 10 to 15% of the cancers, appear to be caused by viruses. It's well known that human papillomavirus causes cervical cancer and skin cancer, and hepatitis B virus and hepatitis C virus can cause liver cancer. There is, not all cancers though, have a correlation with viruses. For example, breast cancer and glioblastoma are not associated with any viral infection. So these two cancers do not seem to have viruses cause them. But other cancers, like I said, about 10 to 15% of cancer can be caused by a virus. Just to tell you, cancer is uncontrolled growth. There are two main ways that viruses can cause cancer. You can have a formation of a provirus, and you can have an oncogenic virus. A provirus is when the virus integrates into your host cell's DNA, making a permanent genetic change in the cell. As I mentioned before, a animal provirus, when it integrates into the host cell DNA, that cell will have that provirus for the lifetime of that cell. That can change the DNA, causing a mutation if you have the gene inserted inside a gene. I have the virus inserted inside a gene. It can change the expression of a gene nearby the virus. It can either turn on a oncogene, or it can turn off a tumor suppressor gene. And so DNA viruses that integrate into the host cell, as well as retroviruses which integrate into the host cell, can cause cancer by the formation of a provirus. The DNA integrates into the host DNA and then just mucks up the host DNA, giving rise to cancer. The second way a virus can cause cancer is when the virus is an oncogenic virus. An oncogenic virus contains a gene called an oncogene, and that's just part of the virus. And an oncogene is a normal cellular gene involved in cell growth, but this normal cellular gene is mutated, and then that oncogene causes the cell to just keep dividing. Is there any question about an oncogene? All right, oncogene is only seen in oncogenic viruses, which is a virus that tends to cause cancer because it carries an oncogene. And when it infects the cell, it tends to cause that cell to just grow uncontrollably, transforming the normal cell into a cancerous cell. All right, any question about the two ways that viruses can cause cancer? Now, we talked about two main ways that animal cells can replicate. or their life cycle. And let's talk about two other ways that animal cells can have their life cycle. The two life cycles we talked about were the lytic life cycle and the budding life cycle. Those are the main ways that an animal cell has their life cycle, meaning how they replicate. But there are two other life cycles, a latent life cycle and a persistent viral infection. A latent life cycle is when the virus goes dormant. The virus goes asymptomatic. It doesn't cause any disease, any signs or symptoms in a patient, and the virus is not actually replicating when it is in the latent life cycle. No disease, no viral replication, and then the virus can leave the latent life cycle. Usually it enters the budding life cycle, and the virus becomes active and starts replicating. In the budding life cycle, you'll get signs and symptoms. An example of a latent virus is the virus which causes cold sores, herpes simplex virus type 1. When the patient doesn't have a cold sore, the virus is in the latent life cycle. It's not replicating, it's just lying dormant. And then the virus can leave the latent life cycle and enter the budding life cycle. And then the patient will get a cold sore. Another example of a latent virus is the virus which causes the chickenpox. A child gets the chickenpox virus, usually when he's young. That's why I said child. And it doesn't have to be. An adult can get the chickenpox virus, in which case the chickenpox is more severe, by the way. But the child will have the virus in the budding life cycle, and the child will have signs and symptoms of chickenpox. Any question about that? Mostly a red, scaly, itchy marks on the skin, although you have a few other um, signs and symptoms too. And then the virus goes latent, and this virus can go latent for decades. And then the virus can reactivate, and it'll come out the second and all later times as the shingles. In the shingles life cycle, the virus is in the budding life cycle, and it's replicating, and that's why it's causing signs and symptoms. Anyways, that's the viral latent life cycle, where the virus actually goes dormant, causing no signs or symptoms, and the virus is not replicating. A trigger is usually what makes the virus leave the budding latent life cycle and enter the budding life cycle, and it's usually a stressor. For a cold sore, what do you think the stressor is? What makes herpes simplex virus type 1 leave the latent life cycle and enter the budding life cycle?
Stress.
Well, yes. What stress? It's actually getting a cold. The stress of getting a cold causes the herpes simplex virus type 1 to leave the latent life cycle and then enter the budding life cycle. And that's why we call it a cold sore, because you tend to get them when you have a cold. Shingles tends to come out of the latent life cycle when the stress is just old age, when the immune system is weakened, and then the chickenpox virus reactivates, causing the shingles. And so the stress is just old age. Your immune system weakens. Any question about the latent life cycle? Let me talk a little bit about the persistent viral infection. A persistent viral infection is a virus similar to the budding life cycle, except it isn't a envelope virus. It doesn't bud, but what happens is we have a naked virus and it gets put inside a vesicle of the cell, and then the vesicle of the cell moves the virus up to the cell membrane, and then the vesicle fuses with the cell membrane, dumping the contents, the naked virus, outside of the cell. and that's a persistent viral infection life cycle. Any question about any of that? Not many viruses follow that life cycle, but it's similar to the budding life cycle in that usually the virus is shed at low amounts. The virus can be shed for long periods of time. The disease for persistent viral infection may occur over a long period of time, and detectable levels of the virus may build up over a long period of time. It doesn't always have to be, but sometimes the virus, let me see if I can do that. See if I can paint out here. Not there. There we go. So usually with the persistent viral infection, the virus will start low and then build up over time. And sometimes it'll plateau, may not get any higher, but other times it will build up over time and then just keep building up. And let's move that out further. And there you'll see it breaks. That breaking could be because the patient died from the buildup of the virus. Let me keep that up just for a minute there. The virus may never be cleared by the host immune system. It may be fatal. In the old days, AIDS, HIV tended to be fatal, and it did build up slowly, and then it bloomed, although the bloom would be much greater here. So let's make HIV something like this. And then the patient would die, and that's why the graph would break. The patient would die of AIDS. Nowadays, that may not happen because of medication that can actually get this viral number to decrease. As far as I know, they never get rid of the HIV. There's been a few recorded cases where they think the patient may have gotten rid of HIV, but most cases, the patient always will have to live with HIV for the rest of their life, which with modern medication can make their life pretty close to the normal lifespan. Where are we? Here we go. Nope, no, we're not. Subacute sclerosing panencephalitis, which actually happens by the measles virus, is a persistent viral infection which builds up over time, and it tends to, let's cross that out now. It tends to kill the patient, so let's break it here. Boy, I'm not able to erase that. Why am I not able to erase that? There it goes. I'm sure no one's ever heard of subacute sclerosing panencephalitis, but I'm sure you've heard of AIDS. In the old days, like I said, HIV acted as a persistent viral infection. It's not. This is actually a budding virus, but it did tend to build up. But many of the persistent viral infection are tolerated, like a patient who has cytomegalovirus, varicella zoster, chickenpox virus, other herpes virus. My mother actually had shingles for the last couple of years of her life because it was always causing the shingles. And She was one of those unlucky patients who, when you get shingles, it lasts more than 6 months. And in her case, it lasted over 3 years. And then she died, not from the shingles, but she always had shingles for the rest of her life. Some patients have cytomegalovirus, I think about 50% of Americans, and it's tolerated. They're always shedding cytomegalovirus at a fairly constant rate, but it doesn't build up beyond a certain threshold. Here's showing you the latent and persistent viral infections. Let me blow that up. Normally, you have an acute infection where from your first infection, short time later, you get your first signs and symptoms. This would be something like the cold. And then the virus goes to very high numbers, shedding very high numbers, and then it drops and becomes extinct in the patient. Acute viral infection, a latent viral infection, something like chickenpox. You'll first have an acute viral infection while the child has the chickenpox, and then the virus drops. You don't see anything at all for many years, and this will be during latency. And then you can have the latent infection when the patient comes down with shingles, which is usually several decades after the child had chickenpox, usually in old age. And that's different from the persistent infection where the virus um, builds up over time, gets worse over time, and it can bloom. Like a good example would be, sorry, I'm trying to remember, muscular, no. Oh gosh, can't remember the name of that disease. The kissing disease. What's the name of that disease? Anyone?
Mono.
Say again?
Mono?
Yeah, mononucleosis is a case of the persistent infection, which does tend to bloom, but this one, the patient usually gets better and then will drop down to zero. But mono is a case of a persistent viral infection, which does bloom. A cytomegalovirus tends to build up and then it just plateaus, but it never fully goes away. All right, let's talk a little bit. Are there any questions about viruses? I guess I should ask that before I move on to the next topic. Any question about any viral topic? If not, let's talk about prions. A prion stands for a proteinaceous infectious particle. They are infectious proteins. They are inherited, meaning you can inherit a prion disease, and they are transmissible by ingestion, by organ transplant or surgical transplant, by surgical instruments. You could theoretically inject it into the patient too. That's only been experimentally done, not into a patient, but into an animal. They cause spongiform encephalopathy. This is vacuoles in the brain in humans and other animals. In animals, we don't call it prion disease. We call it by a specific name. In humans, we call it Creutzfeldt-Jakob disease for the families that tend to have a prion disease run in their family, or Gerstmann Straussler Schnickler syndrome, which is a very similar disease and it's a prion disease that tends to run in the family. Fatal familial insomnia, which I actually don't know anything about, and then Kuru, which I know a lot about, and so I'll talk most about Kuru. But let me show you the spongiform encephalopathy. Here is a normal brain, and these are plaques in the brain caused by a prion. And this is making the brain look like a sponge, why it is called spongiform encephalopathy. Any questions about any of that? This is Creutzfeldt-Jakob disease. That's scabies. This is a disease that ranchers in Europe had been dealing with for centuries. So this disease among sheep is very common. Well, not very common, but familiar with ranchers in Europe. They've been dealing with it for centuries. And then Kuru, the disease that happened in Polynesia. Let's go back. Kuru was a disease seen in the Fore tribe in Papua New Guinea. There's Papua New Guinea, the red region there, the region of the Fore tribe. When patients get Kuru, they become unable to walk. We have the loss of the ability to swallow or chew, drastic weight loss, and eventually leads to death. How was Kuru transmitted? The Fore tribe had an unusual funeral ritual where they cooked their dead relatives and then ate them. And somebody in the Fore tribe came down with a prion disease, and then their relatives cooked him when he died, or her died, whoever, and that caused the Kuru to spread among the Fore tribe. Any question about any of that? Talk a little bit more about the Kuru, and that is the women of the tribe noticed that the Kuru was spreading among people who ate their dead relatives, and so the women largely gave up eating their dead relatives, and they did not allow their children to eat the dead relatives either. And so Kuru was mostly being spread among the men who continued to eat their dead relatives when the missionaries came to Papua New Guinea. And the missionaries were aghast at the Fore tribe where they were eating their dead. And so they essentially made it against the law to eat dead people and stamped out the ritual of eating their dead. And that largely got rid of Kuru in Papua New Guinea among the Fore tribe. And you can say what you can about the missionaries, but this is one time where their intolerance, I think, actually did something beneficial to the natives that the missionaries were administering to. Any question about any of that? That's my opinion, obviously. It's not a scientific fact, but all right. A prion, proteinaceous infectious particle. How did this disease spread? First, you had a normal cell surface protein, which had to be on the nerve cell. And this gene is called PRP superscript C for the normal cell surface protein. And all nerve cells have this gene, which is secreted. Like I said, it needs to be on the nerve cell. And this protein here could come up to the scabies or the prion form of the disease, which is called PRP superscript SC. And the only difference between this protein and that protein is the folding pattern of the amino acids in the protein, meaning the sequence of amino acids in PRP subscript or superscript C and PRP superscript SC is the same amino acid. So it is the same protein. The only difference is the folding pattern of the protein. And then this prion protein would come up to the normal protein and change the folding pattern of the normal protein into the prion protein, the scabies protein. Now the cell has two scabies protein in it and one less normal protein. The cell would say, oops, I need to make more normal protein, and it'll make more normal protein. The trouble is now there's two scrapies protein, either which can come up to the normal protein and make more scrapies protein. The cell will then say, oops, I need to make more of the normal protein and make more of it. And that will make more and more of the scrapies protein accumulate in the nerve cell. As this accumulation occurs in the brain cells, you'll get plaques forming, and eventually enough of the scrapies protein will build up that it will cause the cell to die. So that is how the prion protein reproduces. It just comes up to the normal protein and then makes more of the scrapies protein. With the scrapies protein for humans who have gotten scrapies disease, it's almost always been from ingestion of the scrapies protein. It's thought that the scrapies protein among sheep, this is the disease that prion disease that sheep get. It's thought that scrapies does spread from sheep to sheep. I'm not sure they know how exactly it spreads because you have to have the scrapies protein shedding from the sheep and then the sheep eating it. And like I said, I'm not sure they know how it spreads from sheep to sheep, but it is believed to spread sheep to sheep, and the sheep do not eat the sheep to get the scrapies protein. Any question about any of that? In Britain, there was a case of a prion disease, both in humans and in domesticated cats, as well as zoological cats. And it was because they were taking cow brains and they couldn't sell it. So they ground up the cow brain and then put it on grain and then fed that grain to cattle. So the cattle were eating essentially cow brains. And one of the cattle came down with prion disease, and then that got put on the grain. The cattle ate it, and then they got scrapies disease, and then that just ballooned out of proportion and caused prion disease, obviously, among the cattle. Almost the entire herd of British cattle, I don't know how many, but a large percentage. And about 100 people, or maybe 150 people, came down with a prion disease, and about 150 British cats, either domestic cats or zoological cats. Any question about any of that? All right, enough about prions. Let's talk about plant viruses. Plant viruses resemble animal viruses in many ways. They do cause diseases of economically important crops like beans, corn, sugarcane, and potatoes. One difference is a plant cell is a tough cell wall. Animal cells don't. So plant viruses tend to enter plants through wounds or via a parasite, like a nematode, a fungus, or an insect, which then eat the plants and then allow the plant virus to infect the plant cell. Infected plants can then spread the virus in their pollen or in their seeds. That's it for plant viruses because plant viruses do not infect animal cells. So we're not going to talk any further about plant viruses other than they exist and they cause damage to economically important crops. Let's talk a little bit about viroids. A viroid is a naked RNA molecule with no protein coat. You can think of it as a RNA virus lacking the protein coat. When this naked RNA, called the viroid, gets inside a cell, it will become a pathogen and cause the cell to make more RNA, which will be a viroid, which will then be shed by the cell. Viroids are only known to be plant pathogens. There are no viroids known in animal cells. Do I have the next slide here? Oh, there we go. Here's a normal potato. These are viroids causing the potato spindle tuber viroid, meaning the potatoes tend to be spindle-like, and that's it. Any question about viroids? We won't talk further about them because they do not infect animal cells. They do not cause disease among animal cells, but they do cause disease among plant cells. Any question about viroids? Any question about chapter 13? If not, let's move on to chapter 14. Chapter 14 is a fairly easy chapter, meaning students tend to do better on this chapter than many other. We're going to be talking about the principles of disease and epidemiology. Know the terms of this chapter. Obviously, know the objectives. Let me see if I can blow this up. I've only got 3 here, but if you know the terms of this chapter, you'll actually learn about 50% of the material in this chapter, because this chapter is very important on terms, terminology, the principles of disease and epidemiology. Understand the relationship between the normal microbiota and its host. And 3, understand the stages of disease development. And then 4, understand about nosocomial infections and how to control them. Any question about this chapter? All right, here are 3 terms you need to learn. Pathology, the study of disease. Etiology, the study of the cause of a disease. For example, the etiological agent of anthrax is Bacillus anthracis. And then pathogenesis, the development of a disease. Some more terms: infection. An infection is the colonization of the body by pathogens or the presence of a microbe where that microbe is not normally found. For example, E. coli is normally a part of everyone's microbiota, meaning it's in people's gut. In fact, it helps us digest our food. And when it's in the gut, it doesn't usually cause disease. I'm saying usually because there are some variants of E. coli, like E. coli 0157, 0111, which do cause disease, but most variants of E. coli do not cause disease in the gut. However, when it's growing elsewhere in the body, it can cause disease, like E. coli in the middle ear causes an ear infection. E. coli in the lung causes pneumonia. Any question about any of that? A disease is an abnormal state in which the body is not functioning normally. So, abnormal state when the body is not functioning normally. Few microorganisms are pathogenic, meaning few microorganisms actually cause disease. Some of them can benefit the host, like the normal microbiota. Many of our normal microbiota, not all of them, but many of them can benefit the host. Only a few microorganisms are pathogenic. There are large communities of microbes found in association with animals and plants. These communities are known as the normal microbiota, microbes that permanently colonize the host. Once a patient is born, like the baby is born, the baby tends to pick up Staphylococcus epidermidis from his mother, as well as from other adults that touch the baby. And that normally will permanently colonize the baby, and then the baby will have Staphylococcus epidermidis, usually for the lifetime of the of the host. Any question about that? There are only normal microbiota found in certain regions of the body. Disruption of the normal microbiota host balance appears to be associated with disease and impaired immune function. So this slide is showing you the locations of the normal microbiota on the human body. The skin, the mucous membranes, especially of the mouth, not the mouth, the face, but any mucous membrane has the normal microbiota. The gastrointestinal tract, all the way through, has the normal microbiota. The eyes are a mucous membrane. The respiratory tract and the upper respiratory tract has a normal microbiota. The lungs themselves do not normally have normal microbiota. And that is, we'll talk a little bit, well, maybe we won't do about this in this class, but the upper respiratory tract does have normal microbiota, but the lungs, the lower respiratory tract, do not usually have microbiota. And if you get microbes growing in your lungs, you tend to get pneumonia. The lower urinary and reproductive systems have a normal microbiota in both sexes. The normal microbiota is one where a microbe normally colonizes the host and then tends to stay with the host for about the lifetime of the host. You do not have normal microbiota internally into the body, meaning microbes are not found in the blood, the cerebrospinal fluid, muscle, nervous tissue, bone tissue. And if you do, it's caused by an infection. But normally you do not have microbes inside the body. You can have a transient microbiota where you may have some microbes for days, weeks, or months. But unlike the normal microbiota, the transient microbiota colonizes the host and then goes away. A classic case of transient microbiota was when I went to visit my aunt. She had a Staphylococcus aureus skin infection, and she insisted on hugging both myself and my mother. I brought my mother to see her sister, and she insisted on hugging us. And then the next time I sampled my skin with just growing the microbes on an MSA plate, I saw Staphylococcus aureus growing, and I said, geez, I must have got that from my aunt because I never had that any other time. And I've been sampling my skin for about every term for several years beforehand. I don't know how many years, but let's say many years. And then 3 months later, I sampled my skin and I no longer had Staphylococcus aureus. And every time I sampled my skin after that point, I never had Staphylococcus aureus. So for me, Staphylococcus aureus was a transient microbiota where it was on my skin for days, weeks, or months. And then after about 3 months or less, it was gone, transient. Any question about any of that? And I was quite relieved because if you don't know, Staphylococcus aureus can cause skin infection, meaning it's a human pathogen. And people who have it on their skin are much more likely to get a boil or a carbuncle or a skin disease from Staphylococcus aureus than people who do not have Staphylococcus aureus on their skin. And if you want to know, about 20% of Americans do have Staphylococcus aureus on their skin. About 50% of Americans have it in the upper nasal passages or throat. Not everyone, but about 50% of Americans do. have it in the throat. It doesn't seem to cause disease in the nasal passages in the throat, but it does on the skin. Any question about normal microbiota and transient microbiota? All right. When we look at humans, we find that humans have about 3 times 10 to the 13th human body cells, and humans have about 4 times 10 to the 13th bacterial cells in and on them. This begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much because we do have more bacterial cells than we do have human body cells. Any question about any of that? Here you're looking at some normal microbiota. The orange spheres are normal microbiota in the nasal epithelium. This right there is not a microbe. That's actually part of our villi in the nasal mucosa. And then bacteria on the stomach, that might be Helicobacter pylori, I don't know, but certainly is a spirillum, and Helicobacter pylori does look like that. This is not a microbe, that's actually the lining of a human stomach. And then here we have bacteria in the large intestine. There we have, well, something that's close to being a cocci, one that's a coccobacillus, either right there or right there. And then this is clearly a bacillus. Right there, that's probably undigested food. Right there and right there. Any question about any of that? Oops, excuse me, go backwards. We found some normal microbiota in some surprising places. Like they found that the adult female bladder has a normal microbiota, meaning there are microbes in the adult female bladder. I've always tried to find out if there are any microbes in the adult male bladder, and I can't find anyone who's ever done any research on males, so I just don't know. This was a surprise because people thought that the urine in the bladder was sterile, and actually, if you actually sampled the bladder, that would have told you no, but I'm guessing they thought it was contamination, that the patient had bacteria in the bladder. But after 2014, we now know that females actually have microbes in their bladder. It's known that there is bacteria in mother's milk, and this is true when the mother's milk is inside the mother's breast, meaning inside the breast tissue in the mammary gland. Females can have microbes inside the breast tissue, which then colonize mother's milk. So mother's milk isn't sterile. Actually, the microbes that the mother gives the baby help the baby digest the milk. So the mother's milk being not sterile actually helps the baby digest the mother's milk. I'm not sure why this was a surprise, because they found that the placenta has microbes in it. The reason why this is not a surprise to me is that the female reproductive system is open. The placenta is not even high in the reproductive system. It's, I don't know, midway. And so it doesn't surprise me that there's microbes in the placenta. What's different is the placenta has a unique microbiome that is only found in the placenta. And obviously, you have to be pregnant to have the placenta. And so, after birth, this unique microbiome disappears. Well, wait a minute. That doesn't necessarily mean that it's going to go away. No, the placenta is around only during birth, not birth, but pregnancy. So uh, the unique microbiome goes away after birth because there is no placenta. Any question about any of that? All right. There is a relationship between the normal microbiota and the host. There can be microbial antagonism where the normal microbiota actually compete with the human pathogen. For example, the normal microbiota can prevent the growth of pathogens by occupying the niche that the pathogen might occupy. For example, on our skin, I've got my skin coated with microbes, normal microbiota, and if a human pathogen were to fall onto my skin, there may not be room for the human pathogen to get established, meaning it might have to fall on top of a bacteria and then it won't even touch the skin and it won't have access to the resources needed to begin colonization of the skin. They can compete for resources. The normal microbiota can compete with the human pathogen for resources, denying the human pathogen to get established and colonize the host. The normal microbiota can change the environment of the host, making it less suitable for human pathogens. One example is normal microbiota tend to produce acids, making the environment too acidic for many normal, many usual, I guess we should say, human pathogens. For example, the skin is made more acidic, the vagina is made more acidic, the mouth is made more acidic by our normal microbiota. Or the urethra is made more acidic as also. The normal microbiota can also produce bacteriocins, which are a protein with a very narrow spectrum of action. For example, E. coli that makes a bacteriocin, a protein, will prevent the growth of salmonella and shigella in the intestines, but it will not prevent, well, econo, econococcus, forgetting the name of that organism now. Faecalis, Econococcus faecalis. So if a patient were to get like a salmonella infection, you could treat the patient with bactericin, and instead of it harming all of the gut microbiota, it would only harm salmonella and shigella and any related bacteria. It would not harm most of the gut microbes. And I've often wondered why bactericins weren't used as an antibiotic. And I did read a few years ago where they were experimenting with treating patients with a disease with a bactericin. So think of bactericins as an antibiotic. It's a protein with a similar mode of action, but it's much more specific on what it inhibits. Any question about microbial antagonism? All right. So microbial antagonism is one way that the normal microbiota can establish a relationship with its host. Another way is with probiotics, the consumption of live microbes intended to exert a beneficial effect. Like Jamie Lee Curtis eating yogurt here, is the yogurt has live microbes in it. It can benefit the patient by giving the patient the live microbes in their gut, which actually can help the patient um, digest their food better, as well as they're much less likely to come down with a bacterial infection of the gut, meaning gastroenteritis. But eating the consumption of microbes has also been shown to help prevent yeast infections on the skin anywhere, but particularly in the mouth and in the vagina. You're much less likely to get a yeast infection if you consume yogurt. Any question about any of that? All right, let's talk a little bit more about the relationship between the normal microbiota and the host. Uh, the normal microbiota is living in symbiosis with the host. Symbiosis, all it means is two different organisms or populations living together. So the normal microbiota and the host are in a symbiotic relationship. There are different types of symbiotic relationships, like parasitism. This is when one organism benefits at the expense of the other. Human pathogens form a symbiotic relationship, but it is a parasitic symbiotic relationship where the human pathogen benefits and the host is at a loss, meaning they do not benefit, they get disease or something like that. Any questions about parasitic symbiosis? You can also have mutualistic symbiosis and commensalistic symbiosis. In mutualistic symbiosis, that means both organisms benefit. Like E. coli in our gut, it benefits because we feed it by eating food, and then it benefits us because E. coli helps us digest our food. So both organisms benefit. Commensalism is when one organism benefits and the other organism is unaffected. Now, normally, when we're talking about commensalism, it's very hard to distinguish slight mutualism from commensalism, meaning I've got Staphylococcus epidermidis on my skin, and most of the time I just give it a home. I will feed it so it benefits Staphylococcus epidermidis, but most of the time Staphylococcus epidermidis does not directly benefit me. Most of the time Staphylococcus epidermidis isn't protecting my skin from a human pathogen. Most of the time, not all the time. So most of the time, Staphylococcus epidermidis on my skin forms a commensalistic symbiotic relationship. Commensalism, where one organism benefits, the Staphylococcus epidermidis, and the other is unaffected. The thing is that Staphylococcus epidermidis can compete with the human pathogen, in which case it'll then be acting like a mutualistic symbiotic organism. So the distinction between commensalism and mutualism is kind of fine. Strictly speaking, I only know of one organism which acts commensalistically, and that is in every human eye, we usually have one nematode which swims around in the eye. It's microscopic, so you don't ever see it. Um, there's only one, so enough food falls in the eye for the nematode to eat. The nematode does not do any damage to its host, but it doesn't do any benefit either. So that is a commensalistic relationship between the nematode and the human host. Any question about symbiosis? All right, let's talk a little bit about opportunistic microorganisms. Some normal microbiota can become opportunistic pathogens, meaning they don't normally cause disease in a healthy individual, but under certain circumstances, the normal microbiota may cause disease. These individuals, which normally have the normal microbiota causing disease, are usually individuals with a compromised immune system, like a patient on chemotherapy for cancer treatment, their immune system is weakened, or a AIDS patient, their immune system is shot, so the normal microbiota can grow on them and then cause an opportunistic infection. Any question about any of that? All right, this is the third time we've talked about Koch's postulate. Koch's postulate is used to prove that a given infectious disease is caused by a given microbial species. Koch's postulate starts with you isolate the pathogen in a sick individual, purify the pathogen up, grow it as a pure culture, and then you inject the pathogen into a healthy individual organism. If that healthy organism turns sick, or the bunny is turned on its side here, because the bunny is sick, poor bunny, that's not quite enough to show that this organism causes infectious disease. You then need to find the same organism in this sick bunny that you injected into the bunny. And if you do all of these steps, you've then gone through Koch's postulate to prove that this microbe causes this disease. Any question about any of that? Robert Koch first used Koch's postulates to prove that Bacillus anthracis causes the disease anthrax. All right, any questions on anything? If not, we'll stop here and I'll take up next next Tuesday in the class. Remember, there is no lab. I will be logging in just to answer questions. I'll be logging in for only about 15 minutes. But if you have a question, you can come to the lab and ask. If you don't have a question, there's no reason to come to the lab. You're welcome to, but you don't need to. All right, any questions? All right, I'll see you next Tuesday or in the lab if you have a question. Bye.